Hey everybody, and welcome to The Secret History Living in Your Aquarium. Again, we have a very special guest, Dr. Anthony Mazerol. Hey everybody, and welcome to The Secret History Living in Your Aquarium. Again, we have a very special guest. We're getting Dr. Anthony Mazerol. Hey everybody, and welcome to The Secret History Living in Your Aquarium. Again, we have a very special guest.
We're getting feedback 4 or 5 times over.
Hey everybody, and welcome to The Secret History Living in. All right. I think we killed the feedback. Okay. It's very odd that we're getting feedback because I just have the stream on another device in the room. It doesn't even have sound.
Yeah, I don't know what's happening.
Yeah, that's very odd. Okay, so in any case, hello. Welcome to The Secret History Living in Your Aquarium. We have a very special guest, Dr. Anthony Mazerol. And again, he's here to share his awesome teachings with us, his knowledge. And he happens to have, because of a misunderstanding, he happens to have a really cool thing that is a discus and a tilapia to dissect. And I think I wrote, it could have even auto-corrected, but, you know, we said we're going to do the tilapia and discuss fish illness or whatever. And discus came out as discuss. So anyways, long story short, we have 2 fish now. And so that was fortuitous that worked out as well. But if you guys have any questions, please feel free to type them in the chat as we go. But I'm going to just kind of turn over the show. I'll be the floating voice, the disembodied voice in the background, and I'll turn over the show to you if that's all right, Dr. Mazerol.
That's fine.
All right. And I wanted to tell folks too, please make sure if you wanted to super chat tonight, that's very kind of you, but please take a look at the pinned comment link to the Amazon ornamental, or the Research Center for Ornamental Fish, and please consider donating there instead. I am a very wealthy tycoon at this point, so please help out a different cause. All right, guys. Well, I'm going to disappear, but I really appreciate everybody showing up, and I'll be in the chat as well. And I guess we'll get this show on the road. This is basically free college class, and you know, thank you so much for making this happen. It's very kind of you, and I haven't seen anything like this on the internet, and I think it will be very instructive for people who are just kind of going into this blind, trying to figure out what am I doing what am I even looking at when they, you know, pull a fish out of a tank that has passed away. So thank you. All right. So let's get you up on. There we go.
There you go. So it just happened last night that one of my wild-caught discus that I had in a quarantine tank just went out of control and died last night. So I was able to put him in the refrigerator real quick. And when I read the email this morning, yes, I probably misread it wrong. When it said we will discuss, I read that as a discus. So I thought, I have one, so might as well bring one. So what we're going to do is we're going to first just look at the external anatomy of the fish. That's always the first place you start when your fish dies, just to see, you know, even if your fish isn't dead, you can look at it to see if there's any external parasites. So what we'll do is we'll go ahead and do that right now. So I'm going to switch over to my little webcam that I have. So this is my wild discus. Oh, sorry. A red spotted green discus from Peru. As we know, the Peruvian discus in the areas that I catch them aren't actually native to the area, but they were introduced from fishermen and from a fish aquaculture person who basically dumped them in the river. So I'm going to bring my camera down and we're going to look at different aspects of the fish. We're going to bring them down. Let me go ahead and.
So there's a question in here, which is, if you have a fish die on you and you want to do a necropsy, is it best to refrigerate or freeze or leave at room temp? What's the ideal situation there?
Ideal is probably put under ice.
Okay.
You put it in the freezer, it's going to freeze completely. And then, thawing out a fish always, crystallization forms in the cells. It's not the best thing. If it's in the refrigerator, sometimes it's not cold enough. So the best thing to do is under ice. If you leave it out, it's going to begin rotting and decomposing right away. So you never want to leave the fish out overnight, especially. But under ice is the best. And this fish was under ice since last night.
Oh, wow. Okay.
That's always the best thing. That's
great to know.
So here we can just look at the eye real quick. Which appears to be fogging of the eye actually is just the reflection of the lights here in my laboratory. So the eye looks perfectly okay. There's no fungus on the eye. We move around and look at all the fins. The fins appear to be good. No parasites. Just so you know, this fish was a male. Male discus will have this point on the dorsal fin, whereas the females tend to be much more rounded. And so that's always not 100% sign of a male and a female, but it works out most of the time. It's a lot easier than just guessing. So again, we're just looking at the fins to see if there appears to be any parasites on them. Let me do a much more close up.
Now, I was a little curious about discus, where you find them near Iquitos. Are they, when you find them in a group, are they all kind of a similar color and ornamentation, or does it vary within the group of discus, or is that like a regional thing?
Well, when we catch, most of the time when they catch a discus, they catch them at night, and they're only catching one or two at a time.
Okay.
In a brush pile. For the most part, they're going to be normal green discus without any spotting on their body. So it's the minority of the fish that have the red spots that you can see here on the body.
Oh, okay.
It may be only 1 out of 40, 1 out of 50 fish that will be spotted. And that's because of where they are actually brought in from. So I just want you to know that, this is the same thing I do with my students. We know that both the tilapia that I have here and cichlid are of the family of the discus, and the tilapia are of the same family, Cichlidae. And one of the key features that cichlids have is you can see the lateral line here. The lateral line is one of the ways that fish perceive their environment. There's a jelly-filled pit, and discus actually have a broken lateral line. So you can see another part of the lateral line that is not continuous with this lateral line. And they're one of the only families that actually have that. Most fish will just have one lateral line all the way through their body. And in fact, some discus actually have a few lateral line pores in their tail that is not continuous with this lateral line. So in some ways, they actually have a 3-part lateral line system. So luckily, no parasites. external parasites.
Now,
Go ahead.
now that lateral line, seeing that there are, there's almost like two of them or 1.5 of them on Cichlidae. Does that mean anything as far as the perception of their surroundings? Do we know, like, are they better equipped to feel pressure differentials or anything? Or?
No.
Okay.
No one knows why they have it. It's just
interesting. Okay.
One of the key characteristics. So from this point, I will go into the gills. So we peel back the operculum. In a lot of cases, you can just peel it back, or we can cut it. And then we can look at the gills themselves to see if we can see any parasites. So I'm going to try to come up with this. Of course, it won't do it. Let me lower it all the way down.
It's kind of a nice camera setup you have there. I like that.
It's a $20 Amazon special.
Ah, nice. Is that one of the like microscope camera type things?
Yeah, I can actually show it at the end.
Okay, sure. Yeah.
So one of the things you can do is you can pull out all the gills or just look at them. Now, this one black spot on the gill is not a parasite. It is actually some dirt that transferred from my probe. So no flukes, no gill parasites at all. So the gills are nice and red, showing that they had lots of blood flow to them, and no parasites whatsoever.
So now, if we were to see ammonia burn on gills, what would that? I mean, would you be able to differentiate that in after death?
Yeah, because what you would see is the gills would actually be frayed in here.
Okay.
And they wouldn't be nice, and they'd all be different shapes.
Okay. Yeah.
They'd all be different sizes. So the gills actually look good.
Yeah, very uniform.
Again, dirt. No parasites. So all good here. So now is the part that everybody wants to see, really, and that is let's look at the internal anatomy. Bring this up more.
Yeah, for anybody who's squeamish, I don't know why you're here.
It's not going to be that bad. It won't be bloody So, how to cut open a fish? The best way to cut open a fish is to start at the anal opening here. Let's see.
That would be the cloaca.
The cloaca. And cut through it. So we're going to cut through the pelvic girdle.
So it's almost like gutting a fish for when you're going fishing.
Yes. Now, discus are going to have a body cavity that is much reduced because of how flat they are. So the intestinal tract in their body cavity is really only about right here.
Oh,
whereas
wow.
in many other elongated, normal-looking fish, it'll be all the way along the body.
Oh, wow. Yeah, I guess like a glass catfish or something like that would have a very condensed set of organs compared to, a larger bodied fish like these.
Now, we don't have smell-o-vision, but, fish, their internal anatomy, when you first open, they're going to smell.
Now, I assume you have a lot of experience in this, obviously, and can you tell by the smell if something's wrong with the fish, like when you're on site? I mean, are there disorders or, you know, infections and things that you're like, man, that just smells off, or that's so pungent that it must be something?
No, all you can tell is whether or not the fish is fresh,
Okay,
or not.
okay.
Yeah. Unfortunately, I don't have the sense to smell like dogs. So I'm sorry, this is not the most exciting part for everybody.
Oh no, this is great. I mean, it's good to know how a doctor does this. So
I'm not really a doctor. I just play one.
do a good job.
Yeah.
You had us all convinced. Somebody said, "I'm eating my dinner, so I must not be squeamish."
Okay, we're almost there.
Now, when you're in the outer, you know, skin and muscle tissue there, fascia or whatever else it's composed of, do you find any worms or you know, parasites in that ever, or is it generally?
I never have in freshwater fish.
Okay.
In marine fishes, you will find some occasionally,
yeah,
but not freshwater fish.
I've seen it in salmon just because I've caught so many salmon in my life. But.
Yeah. So let's see. So what you see mostly right here, that is the liver. So most of the fish is going to be the liver that you see. Underneath the liver, if you flap open the liver, you'll see the intestine. So here's the intestinal tract here. I can try to pull it out.
Now, if they have something like fatty liver disease or anything like that, is that something you can see just with your eye, or is that harder to tell?
You can see it, but you're not going to find fatty liver disease in a wild-caught fish.
True, yeah. But if you've been feeding your betta bloodworms 7 days a week for
white worms, it's
2 years,
not the best thing for it. So let's go down and see if we can turn some lights on. Now my camera's going to stink now. Got my fingers all over it. So let's move up here first. So we have a little balloon. Oh, you can't see right here. This is the swim bladder. And we know the swim bladder is used to control the buoyancy of the fish. And so you can see the swim bladder itself actually is vascularized. So these veins actually help put in and take out gases from the swim bladder. It's called a physoclistic swim bladder. There's two different types. Physostomus means that you know, they can gulp air and put it in their swim bladder. Well, most advanced fishes can't.
Oh, interesting. So
go ahead.
Oh, so does that use cell transport, like white blood cell or red blood cell transport or something along those lines to move the gas in and out of the?
It just diffuses in from the blood itself.
Okay.
Yeah. So swim bladder, we look at the intestines again. We'll try to tease the intestines out. And we can try to look inside the intestines. Here, this dark object here, that is actually the gallbladder. It's green because it's filled with bile.
And I would guess that wouldn't smell very good if you ruptured it.
No, the fish itself doesn't smell very good.
Yeah.
So
Somebody says, can you perform this exact same thing on a pea puffer next?
sure. Nice and small pea puffer.
Yeah.
We all want.
How about a micro rasbora, guys?
Yeah. Okay, so this is the stomach up here. So there are some granules in the stomach, and that can be one of two things. It could either be what it's been eating. I feed all mine frozen bloodworms and petrobits. I try not to feed worms as much as possible, just because once you get them on bloodworms, blackworms, that's all they ever want.
Yeah. Now, are they one of the species that will eat stones to help digestion?
No, there's very few stones in the Amazon River.
Oh, okay, sure. Yeah, that makes sense.
Yep. So I'm just trying to pull the intestine out, intestinal tract. So I'm going to pull the camera. I just want you to see how long the intestines are. So this whole thing is the intestinal tract. So the intestinal tract is about a good 8-9 inches long.
Wow.
And so, if you would try to find parasites, you would actually have to lay this whole thing open to find the parasites.
Wow. Okay.
And unfortunately, with this camera, I don't think this camera has the clarity to get down and look at the parasites. Okay, so I'm actually right on top of the intestine.
Now, do you use a scalpel and a probe of some sort for this, like a metal rod, or what do you recommend people use?
So I'm using right now is just a probe, a metal probe. You can use micro scissors, probably the best thing for it, but micro scissors are in Peru right now. So I just find it easier just to slice it open with the probe, and at the same time you can lay it open. So from what I can see, there are no apparent parasites in the intestinal tract.
And now, generally, would those look just like little irregular white or black or gray worm-like things? Yeah. Or, I mean, are there other shapes that we would be looking for, really?
No, you would be looking for worm shapes. You would also, one of the things you could also see is if they're parasites or if they're like tapeworms. The tapeworms are segments. So you could actually see the segments of the tapeworms. And because this was still a fresh kill, you would actually expect the parasites to still be living. So you would see them crawling around as well.
Okay.
So I'll do one more slice. Nope. That one, those two little lines, which you would think are parasites, are actually the blood vessels.
Yeah, I think on our screen it actually shows up pretty well that they're blood vessels.
Yeah.
So that's nice. With the human eye, it might look a little different, less clear.
Yeah. Yep. So luckily,
so they were quarantined well.
They were quarantined, and what we use in Peru, and once I bring them back to the States, we actually use ProziPro.
Okay.
I'm not giving any. I'm not trying to make money because I don't make money off. That's just what we use.
Yeah.
It's easy because it's liquid, and so you can use tin sulfide, which is what we used to use in the old days.
Yeah.
It was a powder, but tin sulfide wouldn't dissolve very well. You'd have to actually use DMSO, and it's just not, it just wasn't an effective tool. So these two lines here were actually the male gonads. So these are the testes of the fish.
That's pretty good size for that fish.
Yeah. But, the testes can be much bigger. These are summer fish. So you would expect them, if they were inbreeding, to be much, much larger.
Okay.
Yeah. So again, liver. Liver looks good. Go back and look in here. This dark object here, that actually is the heart.
So now in fish, are heartworms a common occurrence also?
Not that common.
Okay.
There are some nematode worms that are found in the intestinal tract. There are some in the heart, but not, it's not that common.
Okay. Yeah.
So you can actually see it. This thing is heart shaped, like you would expect a heart to be. Sorry.
Do they have 4 chambers like a human heart?
Yeah. Yep. Okay. So this fish actually looks pretty clean.
Yeah.
So from its symptoms, it started spinning around last night and then basically killed over. Acted normal up until that, so it may be a virus of some sort going on. And if it's a virus, you don't want that to happen, because viruses are very hard to control.
Yeah.
As we know,
Yeah.
as we all know right now.
And so there's not really, other than wasting or something like that, there's not really a good indicator of viruses
No.
after death.
No, not at all. You could take swabs, and try to grow some stuff, or do some DNA sequencing, but for our purpose, even though I do have a DNA sequencer here in my lab, I'm a hobbyist first and a scientist second. I'm not going to spend $500 to figure out what killed my fish.
Understandable.
Yeah. Okay. Anybody want to see anything else in my discus?
We have a request for where are the kidneys?
the kidneys. The kidneys are up in the same general area. They're normally along the back. They're probably have been mixed with the gonads here.
Okay.
So when I pull the gonads out, I probably pull the kidneys out. So the kidneys are going to be two long lines along this area here.
Oh, interesting.
Yeah, they're not like our kidney bean shaped kidneys. They're more of these long tubular, two long tubular shafts that run along the back, right underneath the swim bladder.
So somebody had a question and said, and I think I know the answer, but they said, what about something like a stroke or a brain hemorrhage? Can you tell any of that going on?
No, I have actually never heard of that happening in tropical fish.
Yeah, if as far as I've seen, anything neurological, you can sometimes see by twitching and like drifting and then twitching a bunch, but an involuntary movement. But I mean, I would assume that the size of their brain and any part of their what do you call it? Their neuro system would be a little hard to see, probably.
Yeah.
Plus, once you cut into it, you've kind of compromised the integrity of any tissue that you could tell if it ruptured or whatnot.
Yeah, and it's one of those things that I've never read or heard anything about a stroke or brain hemorrhage in fish unless it has an injury.
Interesting.
So I'm just trying to figure out if that was the food, its last meal. This was actually in its mouth.
Oh, right. So when you're on the way to the stomach, is there like an esophageal tract that's worth checking out too?
Yeah, it's a little one.
Okay.
So what I did is I squeezed its throat and extracted this from the actual mouth. So I brought it up from the esophagus.
Okay. And somebody was wondering, with hole in the head disease and other things like that, are there always ulcers, or is there internal indications ever that, like, before the external ones?
Normally they'll start externally. It's
Okay.
amoeba, so it's you may see some signs of it beforehand, but you're going to have to cut it open to see any signs.
Right. Okay.
And so by the time you see it, the outward appearance, your fish is far along. But the best remedy for that is just water changes.
Sure.
Continue to do water changes, and you should be fine. So I think this was just its last meal. There are some shrimp in there in the tank, so this could be shrimp eggs or something. Or
interesting.
Could be. No, they're soft. They're not.
Almost looks like fatty or something.
Yeah.
Now,
there are some pebbles in there.
Oh.
Discus are known to be detritivores.
Okay.
So that's, they do pick up detritus.
Yeah. Which is probably a great place to find nematodes.
Yep. Okay. That's the discus. Not much internally on it that you can really see.
Yeah.
So we can move on to the tilapia. So just
All
so you
right.
know, I picked up this tilapia this morning at one of our local markets that sells live fish. So I'm right in the heart of Little Saigon here in Orange County,
Oh,
and
great.
there are great fish markets there. You can get live fish from around the world, frozen fish from all over the world. So it's a good place. So these were um, aquacultured fish
Oh, okay.
from Southeast Asia. Live tilapia right now are not coming in too frequently here in California. So I don't know how long this thing has been dead.
Sure.
So I
Uh,
don't know, brought in frozen or what.
So we had another question too, and it ties into something that I've witnessed and been curious about, which is in tetras. Somebody said they have a black skirt tetra, but they have these black, like clumps or set like black patches near the mouth, or I've seen them along the spinal cord in translucent tetras. And they're like a blackening that spreads over time. So I was curious if you know what that would be at all.
I do not know it.
To me, I was assuming it was some sort of tumorous growth or something.
Well, it could be as the fish ages. They do, in some cases, some fish will get an expansion of the melanophores. But I don't know if it's a disease agent at all. So I want you to notice one thing, and this is probably because of the age of the fish in terms of how long it's been dead. But you can see that these gills are not as bright as
No.
the discus, because it was a freshly killed or freshly dead individual, still had plenty of blood in its system, and so you'll see that in this. These tend to be a little bit browner. There
Yeah.
is a disease in fish called brown blood disease, and that happens when there's a lot of nitrogen, ammonia in the water.
Oh, interesting.
So some of the hemoglobin takes up some of the ammonia, and instead of taking up oxygen, the hemoglobin binds to the ammonia, and that causes the gills to turn a brown color.
Oh, interesting.
So gills look good.
And is that milkiness? Is that just
That's
just being dead?
just from being dead. Yeah.
Yeah. Okay.
The slime from the fish is it's all over its body.
All right. Now you can actually see a little more of that redness on the gills too.
And that's because I've scraped away.
Oh yeah.
Possible.
Got it.
But you can see that the gills are nice, uniform. No tearing of the gills, no tatteriness of the gills. So far, looks good.
Thank you for enduring the smell for us.
It doesn't. This smells like happiness to me. So we're gonna go internally on this thing, and we hopefully, because things are gonna be much bigger, we may be able to see a lot more detail.
Sure. And so, is this a Nile tilapia?
Yeah. Tilapia have been hybridized for aquaculture.
Yeah.
So, mixture of a blue tilapia with Nile tilapia, Mozambique, all sorts of things.
I was just looking at tilapia today in preparation for this, and amazed to see how many different both collection points and subspecies or varieties there are of them.
Yeah.
There's some that are really quite beautiful.
Yes.
So what order do these guys belong to?
Order?
Or
Perciformes is the order.
okay.
And family, they're cichlid family.
Okay.
So basically, they're they're distant relatives to the discus.
And is that due to when Earth was all one continent, Pangea, or was that due to fish swimming across the world and then splitting up?
No, it's because of Pangea.
Okay. Right.
Even though tilapia can handle salt water, as we well know.
Yeah.
I'm cutting behind the gills. You can't see. Okay.
So is this kind of a T-shaped incision in theory if you were to symmetrically do it, and then down from the cloaca up towards the belly / gills and the that pelvic fins? Is that what those are there?
Yep, those are the pelvic fins.
All right. And is it easier to cut just to the side of the belly on one side or anything, or is it all as soft no matter where you cut?
No, along the mid ridge of the body here.
Okay.
And then you can cut higher up into here, but then you start getting into the pectoral girdle. So there's more bones here. So I always just do it as close behind the pectoral fin as possible.
Somebody says, can you show us the organ that makes tilapia taste so good in tacos? That would be the muscle and meat.
Yes. I do not eat any farm-raised fish.
I do not either.
I teach sustainable aquaculture, and we could tell you stories about farm-raised fish.
Yeah.
So if I eat anything, it's going to be wild caught.
Yes, I'm with you on that one.
Okay, so that's the tilapia opened up. And in this case, what you see here is fat. So all this white stuff here is fat. And so you can pull it out just like you can pull out like it's butter.
Do not use it as butter, folks.
Oh, tilapia butter is good.
Yeah, that's quite a robust internal organ situation compared to the discus. You can really see.
You can see it's much bigger.
Yeah.
Let's put the fish up against each other. So that's my discus.
Wow.
And the internal cavity of the fish of this tilapia is as big as my discus.
Wow.
And, the discus was a good medium size, probably 4 inches standard length.
Holy cow. Yeah.
Now, does your audience know the difference between standard length and total length?
No.
Okay, so in science, when we talk about fish, we hardly ever talk about total length of a fish. The reason why is, if this fish has his caudal fin bitten off, well, it's going to give you a wrong representation of the overall length. So in science, we do what's called a standard length. And the standard length is from the tip of the snout to a region of the body called the caudal peduncle. And that's, if you bend the fin, the caudal fin back, it's the point at which the last vertebrae ends. So that measurement's called the standard length. And that's what you measure on fish. So you never give a total length of a fish. And so, when people sell fish and say, oh, my fish is 6 inches long, well, unfortunately, they're going to be talking about total length of the fish, but it may not really give you a good idea of the size of the fish. So when I talk about size, it's always standard length, right to here. On the discus, you can basically see it just right where that last black bar is of the discus. Right there is
Yeah.
the caudal peduncle. That's what this area is called, the caudal peduncle. So let's pull out some more fat from our tilapia. So you want to fatten up your fish before you go to market, because the heavier it weighs, the more you're going to get for your fish. So I'm just pulling out all the fat still.
You know, and fish farms are known to do all sorts of odd things, like, well, they're not that odd economically, but use salt to cause things to swell and feed very starchy, cheap foods right before harvesting. And it's not for fish welfare. Let's just put it that way.
No, it's
for
profit.
money.
Yeah, it's always for profit. I mean, you can't blame them for wanting to get the maximum profit. I mean, that's their livelihood.
But you cannot eat it.
Yeah, you can choose to eat it or not. So again, the biggest organ that you'll see inside the fish is going to be the liver. So all of this,
oh wow,
is the liver of the fish. So it has that same liver color that you find in beef liver when you eat liver and onions.
Yeah.
If
now
you eat liver and onions.
Now that laceration there, midsection of the liver, is that just from cutting into the fish, or is okay?
No, I caused that. Yeah.
Okay.
Yeah. So let's see. Someone asked about the kidney.
Mm-hmm.
If you see this kind of off-white line along my finger,
mm-hmm.
that's actually where the kidneys are.
Oh wow, that's very different from human anatomy.
Yeah. And on the other side of the kidney, if I would have cut more, is the swim bladder underneath this chunk of meat here.
Now, do they have two kidneys also?
Yeah, they have a pair of kidneys.
Okay. Is there anything about fish that is irregular from human organs? I mean, as far as the count or having or not having?
No, nope. They have what we have.
Mm-hmm.
I don't think we've discovered an appendix yet.
Yeah, I was gonna say they probably don't eat bark, so that makes sense.
Yeah. The gills. If we go back to the gills,
mm-hmm.
the gills are multi-purpose for fish. Their main function is to take oxygen from the water, bring it to the body. But they're also on the gills. They have cells called chloride cells. And whether they live in freshwater or saltwater, the chloride cells will either take up salts from the water or will excrete salts from the body through these chloride cells.
Right. And this is why saltwater fish and freshwater fish drink per se differently. One doesn't need to drink. So
yes. And it's actually the opposite of what you think.
Right. Yeah. Do you want to explain that briefly?
Well, if we look at this freshwater fish here, it's surrounded by water that, if we look at it, we'll call it almost pure water. So there's more salt inside the body of the fish than outside. So by pure osmotic pressure, the water is going to want to equalize out. So because of that, what happens is water is going to just flow into the body by pure osmosis. Nope, I'm sorry. Yeah, it's just going to flow into the water by pure osmosis because it's want to equalize that osmotic pressure. Well, what happens is the fish then has to get rid of all that water. And when they get rid of that water, they get rid of a very dilute urine. Also,
urea.
Urea has some organic salts. Urea is one of them, which is, ammonia, basically. And so what they have to do is now they tend to have to take in more salt. So they have to, their chloride cells are used to take in salt.
So just if folks are curious, I happen to have a video called "Do Fish Drink and Do Fish pee. So I go over all that in quite a bit of detail with, I think there might even be an illustration in that or two. So if you're curious about that topic and want to know more, because it's pretty fascinating to me that freshwater fish and saltwater fish. So what do brackish fish do? Actually,
Whatever they want to.
Okay. They just kind of pick one or the other?
Yeah, it depends on, depends on the salt concentration of the water. And so, the ones that really do different things are like salmon and other fish that migrate from the ocean to freshwater and back. So they're doing this through hormones. All of it's caused through hormone production.
Right.
So they're going to have to really change their body completely over. So,
yeah. So, and also, just because I've harped on it on my channel before, but there's also something that occurs, which is a rather new study in genetics, which is called epigenetics. Yep. And there are markers or tags within the genetic sequence. So just like a human can go through puberty, but your DNA stays the same, it turns off and on different cues in your body. And fish do the same thing when spawning or going into salt or fresh, large changes in their life or travel cycles.
So let's look at the intestinal tract here. So we'll start up on the stomach is here. It's an enlarged area of the intestine, really.
Yeah.
It's more of a collecting site and starts the digestion. If I pull up here, this intestinal tract is, let me wrap it around. It's about, there's the
oh, wow.
And there's the intestine. So I've wrapped the intestines around. It's about 4 times the body length of that fish.
Wow.
One thing, if you don't know what a fish eats, let's say you discover a fish and you have no idea anything about the habitat, where it lives, or what it eats, look at the intestinal tract. If it has a very long intestinal tract, 5-6 times its body length, that means it's either an herbivore or an omnivore who tends to eat more plants. A short intestinal tract is a meat eater.
So is that due to kind of the same effect of, cows needing 4 chambers versus 1?
Oh, yeah. What
fermentation.
That plant material is so hard to break down that you're going to need a long intestinal tract to break down that plant material and to absorb all the nutrients. Now,
right.
There's ways of overcoming that. I worked on a fish, brown surgeonfish, and brown surgeonfish have bacteria in their stomach.
Right.
In fact, at one time, it was the largest bacteria ever known to mankind.
Really? That's interesting.
It's been surpassed now by a bacteria in Yellowstone.
Oh, that makes sense. Yep.
Yeah. But so it's the bacteria that do all the breakdown of the starch materials. So the fish is just a housing for these bacteria.
And in some cases, you kind of see essentially fermentation occurring too, that changes the composition of the food, or enzymes that can almost change proteins and different things to the point of cooking or heating wood. So the body can do a lot of incredible things depending on the fish and how it evolved.
So let's look at the intestines again. I opened up the intestines.
So somebody said, so if you have a freshwater versus a saltwater fish, are the chloride cells the main difference anatomically? Or is there, some organ or anything important that you can tell, like, oh, that makes it a saltwater fish versus fresh?
No, it's the chloride cells are almost anatomically similar. They're going to be the same. It's just the function of the chloride cell.
Interesting. So I wonder if you could easily engineer with epigenetics the ability of freshwater fish to process saltwater.
Well, most freshwater fish are not going to have a problem with saltwater. It's the other way around. It's saltwater fish that are going to have a problem with freshwater.
Okay, yeah, I guess, yeah, I see what you're saying. Yeah, that makes sense, because they already have to regulate sodium and yeah, that makes sense. Okay.
So here on this fish, this paired organ here, again, these are the testes of this fish. So this is a male. So these are the male organs.
We got 2 males tonight.
Yep, just happened to be what they have. And again, in the reproductive season, this organ would probably be twice the size.
Wow. And so scaling wise compared to oviducts, what's the difference there? I mean, so
the ovaries are going to be probably 5, 6, 7 times as large.
Okay, all right, and hence why you can make out the difference between a male and a female in a lot of fish.
Exactly.
Just due to size.
Just due to size, and you can actually see the ovaries. The organ itself is going to be glandular in a female.
And from what I've been able to find online and in one old textbook that I have, it seemed like the reproductive system of fish was a lot less complicated than humans. Like they say oviducts and then ovitracts, and it's just like no fallopian tubes, obviously no womb or anything. Live bears are set up a little differently, but it's fairly primitive as well.
Yep. So
ova meaning egg. Sorry, guys.
Yeah, ova is eggs. If you give me 2 minutes, I can go get some ovaries and show you what ovaries look like.
Sure. I mean, if you got a supply of ovaries hanging around,
I have a supply of ovaries here. Just have to go get it in the cabinet.
No problem. No problem.
So talk amongst yourself, and I'll be right back.
Thank you so much. All right, guys. Well, how's it all going? Mick, we covered the hole in the head thing, and basically the ulcers are in the head, but they can be under the skin or in the inside of the tissue, but there's not really a good way to figure that out, without cutting open the fish, and then it's dead anyways by the time you've done that. But if you're checking it out, that won't have killed it. It may have that subsequent to it, but that doesn't necessarily tell you much, because the stress that's already been imposed on the fish is pretty, could have already caused other issues. Yeah, no problem, New Local Austin. I appreciate everybody coming out here. Thingamatorium. So what is the reason besides oxygen content fish die from warming temperatures? It's like global warming and food supply. Well, so global warming is going to be a much different phenomena. You're going to have a change in everything, from the bacteria that are present to the food and grass that grows to where the fish want to be for optimum egg temperature and things like that. Just like in humans, also sperm, or in fish it would be called milt, but it needs to be stored at a specific temperature, ideally, for maximum motility. And so it can be controlled actually within that gland. They can control its temperature via some sort of I guess it's a liquid that regulates it. I don't know if it's certain cells or what. But so that's kind of like an overall climate issue versus a localized issue. But if your fish was in a hot tank of water, it would die from a lack of oxygen. And then you would see The gills may not be as red, I would assume, but now that the doctor is back, we may be able to ask him as well. If there's any way to tell if a fish died of, lack of oxygen in the water or heat or something.
First thing I would do is check the temperature.
Yeah.
If it's hot and they're dead, they've died from temperature stress. It may not even be from lack of oxygen.
Sure.
It may be that their body is meant to. The enzymes of the body and the organs of the body are meant to work at a certain temperature range. And if you get above that range, your organs start to fail. You go into organ failure. So it may not necessarily be oxygen stress, because all fish have the ability to come to the surface and gulp air.
Right.
And that's the first sign that you have a hot tank. Or there's something going on with the oxygen, is if they're at the surface gulping air. It may be that you have a slick on top of your tank that's not allowing oxygen exchange.
Too much duckweed.
Yeah, too much duckweed, that can cause no exchange for the atmosphere and other things.
And while we're on it, just a property of rather a physics issue rather than an anatomy or chemistry issue. Water, the gas exchange occurs at the surface area where it breaks in water. So that's going to be at the surface. You want surface agitation to increase water, which then allows evaporation, which is essentially room temperature boiling or boiling below the sublimation point. And that will occur no matter how hot it is. If you can keep the water moving and expose the most surface area and volume to air as possible, it will cool down and evaporate, and that body of water will stay cool much longer. But so if it's a real hot day, you want to put fountains or even a power head or something in that breaks the surface tension, rather than, you don't need an air stone necessarily. The air stone works because it breaks up the surface tension, and some gas bubbles do diffuse into the water, but at about 90% less effective rate as when it breaks at the surface, because water is a polar molecule.
Yeah, so. Your air stone has to create micro bubbles for that oxygen to get into the water.
Right.
So really, all the air stone's doing is breaking the surface tension of the surface to allow that exchange between the water and the air itself.
And moving the water slightly as it pulls towards it. Yeah.
It allows for circulation. So we'll go back to our gonads. You can see here how glandular it appears.
Oh, wow. Yeah.
And all these, that gland, what you're looking at, each one of those little bumps is a fish egg. So
now, what fish is this? Is this a tilapia also?
No, this actually came from a perch, a yellow perch.
Oh, okay.
So they probably have a good 10, 20, 30,000 eggs here.
And are they born with fish. Are they born with eggs like humans, a set number, and then they only develop in rotation, kind of?
No, they are born with, if it's a non-sex changing fish, they're born with either ovaries or testes. Primary, they both first have primary, and then as they mature, then they will start producing eggs.
Okay.
And there is a relationship. The larger the fish, the more eggs it's going to have.
Yeah, that which is really interesting. There's some set rules, and I can't remember the name of the professor who figured it out at the turn of the 19th into the 20th century. But also, the mass of an animal determines its heartbeat and its body temperature, and there's some interesting connections that are your general rules that are true.
Okay.
Wow.
We want to see anything else?
All right, guys. Do we have any other things we'd like to see? Perhaps the swim bladder in the tilapia, if that isn't too much trouble.
Yeah, I actually poked it as I was.
Oh, okay. No,
No. I can get it. So yeah, I was playing with a tilapia while you guys were talking, and gotta amuse myself.
it's like a bubble wrap or something.
Yeah, just putting on new gloves. So go. Where are we at? So let me cut into this.
So, I mean, this was a pretty disease-free tilapia, really.
Yeah, which is surprising.
I'm surprised. I was kind of expecting to find nematodes.
Yeah.
Or flatworms somewhere.
Somewhere, yes.
And maybe, I guess, if you kept looking, you might find them somewhere. But
yeah, if I had a actual compound microscope here that I had a camera on, you could probably see them. But so we're going through the rib cage here. So you can see that hole that I just made this hole. But here is the swim bladder. So
interesting.
Much bigger than the swim bladder for sure in the discus.
Now, are you cutting away like some sort of fascia or tissue that
surrounds that's all muscle tissue.
That's all muscle. Okay.
Yeah. So it was actually underneath. So if I lay the flap, is underneath this big flap of tissue here. So I'll show you how thick it was. So that's a nice thick mass of muscle.
Wow, yeah.
It's underneath that muscle.
And I know from experience that is not always there in small fish.
No. And again, the upper body cavity is lined with fat too.
Yeah, that silvery.
No, there's this stuff that I'm pulling away here. It's not actual muscle tissue, but fat. So that silvery area here is just the swim bladder itself.
And again, we see the veins on it.
Yep, you can see the veins.
So it's the same type of swim bladder as the discus.
Yep.
And so can you maybe explain to us just really quickly what the swim bladder does for a fish?
The swim bladder will help regulate the buoyancy of the fish. If you're a fish and you're in 100 feet of water and you're a mid-level fish, you don't want to have to continually swim to stay at that mid-level. So when they stop swimming, they actually will just stay there. They don't start sinking. If they do start sinking, then they'll pump in more gas into their swim bladder. So it'll keep them just neutrally buoyant in the water at that level. Now, when we have deep sea fish, when people reel up deep sea fish that are thousands of feet below the surface, when they come up, their eyes are bulging, their intestinal tract is hanging out their mouth. That's because they bring them up too fast, and the swim bladder does not have enough time to equalize.
Okay.
So the swim bladder expands exponential until the point where it just forces all the tissues out of the body. There are remedies for that. You can put a hypodermic needle into the swim bladder.
Oh, right.
If you're bringing a fish up for 300 or 400 feet and you're actually doing it with yourself scuba diving, there are scuba rigs that will allow you to go that deep. You can put in a hypodermic needle, and as you come to the surface, air will just be expelled out of that swim bladder, and so it'll come to the surface without any problem.
We've actually seen that salmon fishing with depth riggers.
Yeah, with down riggers, yeah.
Down riggers, yes, exactly. Cod and halibut as well.
Yes.
Yeah. So let's see here. So what system is the swim bladder a part of in a fish? Like, somebody asked, is it part of the limbic system or?
Yeah, I call that too. It's part of the swim bladder system.
Okay, yeah, it's kind of a unique function.
Yeah, It's
I would assume it has connections with neurological things and vascular things, but but it's its own organ.
It's its own organ, yes.
And so I recently did a video update from a store where we looked at lungfish, and they just recently figured out that lungfish came from that their lungs were not evolved from the swim bladder. It was the other way around, which I thought was interesting. Or rather, they had lungs, and the lungs didn't turn into the swim bladder evolutionarily. They don't think in that fish.
Yeah. Oh, okay.
Which just surprised me. I figured.
Because the thinking was that your lungs, developed out of especially the physostoma swim bladder. The swim bladders that have a connection to the esophagus, so they can belch out air or come to the surface and suck in air.
And so that's like when you see Corydoras zooming up to the surface and sipping air. So the other thing is the intestinal system in cories and some fish. It states, in the literature that it's their colon, even, which surprised me. But that they can absorb air, oxygen through their digestive tract, and it just depends on where. It depends on the fish.
Yep.
Richard says, is that why when you catch a bottom fish, it's hanging out of the mouth? Does that kill it right away if you throw it back?
Those fish are dead.
Yeah.
Yeah.
So do they get the bends also, or have they evolved to not worry about the nitrogen?
Yeah, we get the bends because we have a lot of spaces in our body, a lot of air-filled spaces. Other than the swim bladder, there's really no air-filled spaces in a fish. And so they don't have to worry about that nitrogen absorbing. We have nitrogen because we breathe it from the surface. It's
70%.
78% of our atmosphere is nitrogen. And when you're on a scuba tank, all they do is they force in air, atmospheric air into that tank. So your body is super saturated with nitrogen.
Right. Interesting. Somebody has a question. Jerome has the question. Do you have a recommendation for a microscope for a hobbyist by chance?
I would stay away from the cheap ones, which you're not really looking necessarily for. The optics are what you want. You want something that has nice, clean optics. That that one looks good.
This is from 1978. I got it, or my friend got it from a middle school, had an auction. Mm-hmm. And it's totally viable. I just bought a little piece, and I can hook my phone or a camera up to it. Yep. But it goes up to 400 times.
So you
can get an old one, and they're better.
You can get cheaper models just as long as you can change the objectives in it to get quality objectives. Because what happens is they're all great at low power, but when you get to high power, you can't really focus in. It's like having, bad glasses. You can never really focus in because the glass isn't made for your eyes. Well, that's how bad microscopes are.
Yeah, and the digital ones are notorious for going in and out, in and out. Yeah. The cheapo electronic digital ones, those aren't really microscopes. They're more like very zoomed in cameras that are fidgety.
So I said I would show mine.
Oh, yeah, right.
This is mine.
I have the same one.
Yeah. It does its job okay. even though it says it has a 1,000 times zoom, it really doesn't have a zoom.
No.
It has to stay, it just brings something in focus. And that's what I was
It has
hoping.
like,
yeah,
it has like 16 megapixels or whatever, and then it basically does a digital zoom on that set screen, which is what most of those do.
Yeah.
It does not have a high quality, Zeiss lens or anything in it.
No. But to see basic stuff, it's fine.
Yeah, and it gets in little spaces.
Yeah, I would never, take a photo with that and have it on a scientific paper because the resolution is not going to be there. So,
yeah. Well, does anybody have any last questions on fish anatomy for the night? And also, if you're curious about terminology, if you're trying to, maybe you're a student and you want to get more involved in this field, and you don't want to look silly, you want to know posterior, anterior, and all the cardinal directions in anatomy and biology, and the organs and the caudal peduncle and all those kind of things. Cloaca. I have 2 or 3 different videos on external, internal, and what was the other anatomy? Oh, skin anatomy or tissue anatomy, which is pigmentation and chlorophylls and things. Would a stereo microscope work better? Somebody wants to know.
It's what I don't like using stereoscopes because my eyes. Well, now would be fine, but before I had eye surgery, when I had the detached retina, my eyes were so different from each other that it was hard for me to use both eyes when I looked into a microscope.
Yeah.
I was raised on a one ocular microscope, so the stereoscopes, they don't. I'm still not used to them. We have them here at the university, and that's what we have in the research labs. But I always just look at them with one eye anyway.
Yeah, and I think if you're doing work for 10 hours straight, then it is better for your eye to use the stereo ones. But I mean, if you're just spending a half hour on the microscope, you'll be fine with either way.
Yep.
Archaeology, I had to look at the edge of a lot of stone projectile points with those things for hours and hours. So looking for fibers and micro abrasions. So a lot of people have just reiterated, though, thank you so much for showing us this and spending your time this evening.
No problem. Can I share a screen?
Oh, yeah, please do. You should have a little share button, and then I can just allow it as soon as you pop it up.
So is it showing my webpage?
Yeah, right. Let me get me off here. Do you want to take up the full screen?
Sure.
All right.
We're having our end of year pledge for money. We're actually at 69% of what we wanted to raise. So if you're interested in donating to the research center, again, we are a 501(c) nonprofit. All of the donations are tax deductible. So you go to our webpage, www.amazonresearchcenter.org. This is from our homepage, and all you have to do is hit the donate button, and that will take you to a donation site that we use, and you can donate $1, $5, $500, $50,000, $500,000.
You'd even accept $1 million, I heard.
I'd accept $1 million. If you donate $500,000 or more, it'll be your research station. But we live and die off our donations. It's because people like Alex and other people and people who watch the stream that donate their $5, $6 that actually help us in the long run. So if you feel like donating, we know this is the holiday season and money's tight for a lot of people. A lot of people are out of job, so that was, my hopes that at some time in the future, if you feel like giving, There are a lot of good organizations out there. We're just one of the organizations, so we would appreciate you just thinking about us for donations.
And if for selfish reasons, folks, I mean, what a better way to there's not a better way to introduce yourself to the Mazerols than make a donation. And maybe if you're headed down to go check out the Peruvian Amazon, like he said in the last visit, they've got dorms that people can utilize, and grad students, researchers, other professors. If you're in the audience, definitely contact him through that website that's linked, both pinned in the comments and it's in the description of the video. But he is open to all sorts of different programs with the public and with Americans or folks from Europe abroad coming in and staying at the center, helping out at the center, or going collecting with that as the base. So just inquire that way. But I mean, you're going to get some favoritism maybe if you drop a couple bucks,
or mention Alex's name, and then I will make sure that you get to stay in the slum areas and not the good parts.
That's right. Yeah.
But I am going to Peru on the 26th of this month, and I'll be spending all of January there. So for those who follow my YouTube channel, it's not a live stream like Alex. I will be posting updates of the aquarium on that YouTube channel probably mid-January, depending upon our internet strength there.
Great. And I'll make sure to relay, obviously I'm not going to rebroadcast, but I'll tell folks when there is content there. Just check in. Problem. We'll cover that on the live stream, folks. Okay. Amongst other people like Ivan Mikoji and friends of the channel, essentially, we try to keep folks updated on what their nonprofits or their research and things, what's going on. So, so I just see a question in here, and it says, can you dissect a frontal lobe? Well, I mean, if you had a really good microscope in a fish, or a giant fish, no, you'd still need a good microscope.
Yeah, but if I had better tools here, the problem with on these fish, the bones are pretty hard, so you need a small bone saw. On a shark, it's really easy because it's all cartilage. But I don't have all my dissecting kits here. I'm not I'm a field biologist, field conservation biologist. I'm not a taxonomist type. I try to spend as little time in the lab as possible. But, you do have to do some lab time in your life. So I do not have the tools to actually do. If you had a hacksaw blade would be great. I don't have one in my lab right now.
I use a Dremel the few times I was curious. That gets messy though, but but yeah, thank you so much, Dr. Mazerol. We really appreciate it, and this was a really neat experience. I've done postmortems on fish, but never. I never knew if I was going into it the proper or effective way, and you really shed a lot of light on the issue for us. And I promised the channel I'd find somebody who knew what they were doing one day, and that day has come. So thank you so very much for that.
And glad I can be of some service.
And again, guys, if you have questions or an interest in visiting the Amazon and helping out down there, or just checking out, he's going to have a public aquarium open down there with native fish, any number of the 300 plus species of ornamental fish found near Iquitos. Please write him an email and get in contact with him. This is, even if you're watching this after the fact of it being live, it's a really cool project him and his wife have going. So I highly recommend you guys check it out. And also, if you have a company or something, or you work for somebody who's looking at giving money to a nonprofit that's worthy, definitely tell them to check this, check out the website, check out the work they're doing, because it's really cool. They're teaching, conservation and preservation on the river to the native folks as well, and children, as well as possible reintroduction of species seized from customs, and all the way up to new sustainable methods of aquaculture, which both helps the indigenous people in the off season when normally they can't go collect, and it helps it can help everything from food sources to income sources. So really what they're doing there touches the entire community in a meaningful way, and it's a good way to help people and animals and nature all at the same time by supporting an organization like this one. So I just wanted to end on that note, and I really appreciate everybody tuning in tonight. This concludes the event for the evening, and thank you so much again for spending over an hour with us on these dissections. So I like to leave the last words always to our guests. So if you had any last words, goodbye everybody, and thanks for coming out. But I'm going to leave the last words to Dr. Anthony Mazerall, and thank him one last time for spending the time and energy and skill and years of expertise here with us tonight. Thank you.
Well, since we're on the idea of fish health, the best way to prevent fish from having diseases is get to know your fish. Watch them. When you see something out of the ordinary, at that point, then you can go in and investigate. But I'm not one who, as soon as I see something in a tank, I want to dump in tons of chemicals. That's what we're doing more for our fish by doing that. All fish have parasites. All fish carry viruses. It's just when their immune system cannot handle that they will get sick. So if you have good water quality, that's 95% of your problem solved right there. And that doesn't always necessarily mean water changes. That could be new biomedium, carbon in your filter. Even though I don't like carbon, but there's other ways of preventing disease outbreaks. So it's best to be proactive than reactive when it comes to your fish health. And that's my lasting thought for tonight. But thank you very much. I appreciate everybody's attention. I hope you got something out of it. It wasn't the best with my camera, but a lot of you have never seen the insides of your fish. Now you have. So now you know what to expect. Thank you.